Good afternoon, everyone. Today we will discuss the topic that is the respiration in plants. Respiration in plants is the plant physiology topic of class 11, and this topic is mainly for the NEET aspirants, right? Now, first of all, if we describe the definition of respiration, it can easily be described as respiration is nothing but a process in which all the food materials are broken down to release the energy, right? And now, which form of energy is it? It is nothing but in form of ATP. And we all know that ATP is the main energy currency of the cell. The full form of ATP is the adenosine triphosphates, where the 3 phosphate molecules are present. So the released energy, which is actually released due to respiration, is not used directly, right? Rather, this energy is trapped in form of ATP, which is utilized for the different metabolic or different kinds of biochemical, metabolical, physiological and cellular processes of the cell. And now this process of release of energy is never be a single step process, right? And it involves, rather there is an involvement of multi-steps process, and each and every steps are catalyzed by different enzymes. So at the end of definition of respiration, it can be said that respiration is a enzyme catalyzed multi-step process where all the food materials are broken down or oxidized to release energy in form of ATP. And the food material which is utilized during the process of respiration can be divided into 3 main categories. And first of all, these food materials involved in the process of respiration are referred to as a respiratory substrate. And now those respiratory substrates are classified into 3 different categories. First of all, the carbohydrate, the second one is protein, and fats, organic acids and many other different respiratory substrates are there, of which carbohydrates are the most simplest one, and carbohydrates are the type of respiratory substrates which are utilized first because of their simplest nature, simplest chemical nature. And the other respiratory substrates like proteins, fats, organic acids are oxidized subsequently. That is, the carbohydrate is the first respiratory substrate which is oxidized from the very early process of respiration, followed by protein and fat. Now the next topic is the breathing versus respiration. Actually, if we say about the breathing is basically nothing. That is, we all know that it involves the inhalation as well as exhalation. Inhalation, that is the intake of O2, as well as exhalation is the release of CO2. Okay, now if we differentiate breathing from that of respiration, it can be easily said that breathing is a type of mechanical process, whereas respiration, as it involves the breakdown of food materials releasing energy, so respiration is a type of cellular process. Breathing is inhalation and exhalation, whereas respiration is a process in which ATP is formed. Now, our next topic is the respiration in plants versus respiration in animals. That is, what is the basic difference in respiration in between the plants and animals, right? So we all know it is obvious that in case of animals there is a presence of specific respiratory organ, whereas in case of plants do not have any type of special respiratory organ. There are many different reasons behind it. The first reason is that in case of plants, rate of respiration is very, very low. And the second main point, due to absence of respiratory organ in plants, is the that the every plant parts, that is the root, stem, leaves, etc. and etc., are able to respire independently, which is actually absent in case of animals. Therefore, the need of transport of gases between the different plant parts, that is the transport of gas from roots to shoot or shoot to leaves or leaves to stem, is very negligible, because the every plant parts that I say in the second point, that is the root can respire separately, leaf can respire separately, as well as stem can respire separately, right? Now, the respiration, respiration is basically of two different types. The first type is the complete respiration, and the second type is the incomplete respiration. In case of complete respiration actually occurs in presence of O2, whereas incomplete respiration generally occurs or always occurs in absence of O2. Here in case of complete respiration, complete oxidation of respiratory substrate take place, whereas in case of incomplete respiration, as there is an absence of O2, the respiratory substrates are incompletely oxidized. Similarly, in case of complete oxidation, as the respiratory substrates are oxidized completely, so there is a amount of released energy is much more compared to that of incomplete respiration. And as the complete respiration occurs in presence of O2, it is also termed as aerobic respiration, whereas the incomplete respiration is generally referred to as an anaerobic respiration. Now, in case of aerobic respiration includes the three different steps, that is the glycolysis. The first step is the glycolysis, the second one is the Krebs cycle, and the third one is the electron transport system, that is we shortly referred it as a ETS. Whereas anaerobic respiration includes the two different processes, that is the glycolysis and fermentation. Among these two processes, that is the aerobic and anaerobic respiration, glycolysis is the universal pathway, that is it occurs in both the type of respiration. Now, the next topic is the place of occurrence. Place of occurrence in case of glycolysis, Krebs cycle and ETS, glycolysis occurs in the cell cytosol, whereas Krebs cycle occur in the mitochondrial matrix, and the electron transport system occurs in the inner mitochondrial membrane. Now, suppose it is a cell with a prominent nucleus having a well-developed mitochondria, and this is the inner membrane of mitochondria. Glycolysis occurs in the cytosol, mitochondrial matrix. Within the mitochondrial matrix, Krebs cycles takes place, and the inner mitochondrial membrane ETS takes place. Now, we discuss the topic of glycolysis. The topic glycolysis or the term glycolysis involves the two different words, that is the glyco and the lysis, of which lysis referred to the breakdown and the glyco means the glucose. Glyco actually refers the term glucose and the lysis is the breakdown or the splitting. Therefore, it can be said that glycolysis is a process in which the breakdown or splitting of glucose molecules take place. That is, the breakdown of glucose is known as glycolysis. The term glycolysis is basically given by the three prominent scientists, that is the Gustav Emden, they are the Gustav Emden, Otto Merhoff and the J. Parnas. And according to their name, the process of glycolysis is termed as EMP pathway. And the entire process of glycolysis, when we describe it briefly, it is seen that the process of glycolysis is a 10-step enzyme mediated process divided into two major phases, that is the preparatory phase and the payoff phase. Now, we discuss the first phase, that is the preparatory phase of glycolysis. Preparatory phase of glycolysis. In preparatory phase of glycolysis, at the very beginning, we must know that what is the form of reserve food in case of plants. What is the reserve food material in case of plants? Plants actually able to store the food in form of sucrose. This sucrose is broken down into glucose and fructose with the help of a specific enzyme known as invertase, and these glucose participate in the first phase of glycolysis, that is in the preparatory phase, where the glucose is converted into glucose 6-phosphate with the help of an ATP molecule. Now this glucose 6-phosphate is converted into fructose 6-phosphate, and this step is the reversible step, and from these fructose 6-phosphates there is a production of fructose 1,6-bisphosphate with the help of an ATP molecule, that is ATP is broken down into ADP plus PI. All those molecules, that is glucose, from glucose to fructose 1,6-bisphosphates, all are 6-carbon compound, and the fructose which is produced due to breakdown of sucrose is also can participate in the process of glycolysis, directly converting into fructose 6-phosphate with the help of an ATP molecule. And the enzymes for the first step of glycolysis is the hexokinase, for the second step phosphoglucoisomerase, and the third step of glycolysis is catalyzed by phosphofructokinase. Now from the fructose 1,6-bisphosphate is now converted into two different molecules. As the fructose 1,6-bisphosphate is a 6-carbon compound, it is now being splitted into two different molecules of 3-carbon compound, of which one molecule is the glyceraldehyde 3-phosphates and the another molecule is the dihydroxyacetone phosphate. Both the molecules are 3-carbon compound and they are interconvertible to each other, and the step is also reversible steps catalyzed by the enzyme aldolase. The second phase of glycolysis is the payoff phase, in which is started from the glyceraldehyde 3-phosphates. At the very beginning, the glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate, and it is a reversible steps catalyzed by the cofactor NAD. As in the payoff phase, there is an involvement of two molecules of glyceraldehyde 3-phosphate, so there is a production of two molecules of NADH in the first phase of payoff phase, in the first step of payoff phase. And from this 1,3-bisphosphoglycerate, there is a production of 3-phosphoglycerate, which is also a 3-carbon, and this step causes the production of two molecules of ATP. And from this 3-phosphoglycerate, with the help of a reversible reaction, it is converted into 2-phosphoglycerate, and these 2-phosphoglycerate is now being converted into the two molecules of phosphoenol pyruvate, and by causing the release of two molecules of H2O, and all the molecules are 3-carbon and the amount is the 2 moles. And from this phosphoenol pyruvate, in the last step of glycolysis, the phosphoenol pyruvate is converted into pyruvate with the help of 2 ATP molecule, causing the production of 2 ATP molecules, right? And each step of payoff phase is basically catalyzed by different enzyme. The first step is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. The second step is catalyzed by phosphoglycerate kinase, and the third step of payoff phase is catalyzed by phosphoglycerate mutase, and the fourth step of payoff phase is catalyzed by the enzyme enolase, and the last step of glycolysis is catalyzed by the enzyme pyruvate kinase. Now the very important topic is the net gain of ATP. We observe that in case of preparatory phase of glycolysis, the first phase and the third phase involves the involvement, or rather the consumption of ATP, that is the -2 ATP, and whereas in case of payoff phase, the last step and the second step causes the formation of 4 molecules of ATP, and the first step causes the formation of 2 NADH. So the net gain of ATP from the glycolysis is the 2 ATP molecule, and the 2 NADH are the direct gain of glycolysis. Now we all know that 1 NADH is equals to the 3 ATP molecule. So from the entire process of glycolysis we got the 2 NADH molecule. So 2 NADH means there is a production of 6 ATP, right? So the net gain of ATP from the glycolysis is the 6 plus 2, that is the 8 ATP molecules. So the direct gain of ATP from glycolysis is the 8 molecules of ATP, and the indirect gain is the 2 ATP and the 2 NADH molecule.